Okay, and so let's take a look at your activity 19, that your specific tests for your amino acids. Okay, and so of course, you have here, we already did your test for your proteins, the properties, and your general test for your proteins. And so now we're going to move on to your specific tests for your amino acids again. And so as what we said last time, and so previously, that your amino acids, these are your building blocks of your proteins. And so meaning when you hydrolyze, and so when you break down your proteins, and so it will give you your amino acids. Okay, and so when you join amino acid plus amino acid and then amino acid and then another amino acid, and so more and more of amino acids, you join them together, you will make a protein. And so that is for your amino acids. And so let's take a look at your hydrolysis of your proteins first. Okay, and so your hydrolysis of your proteins, and so your hydrolysis of your proteins with your dilute acids, alkalis, or enzymes, and so will produce fragments of gradually diminishing complexity. And so like they are the hydrolysis of your proteins, and so it is also is like also for your carbohydrates and fats, no? And so again, you have there when you hydrolyze, and so you produce fragments of gradually diminishing complexity, and so they become less and less complex until of course you produce dipeptides, and then finally to your uh, individual alpha amino acids. Okay, okay, and so you have here, and so when you hydrolyze your proteins to your when you hydrolyze your proteins, and so when you subject them with your dilute acids and your alkalis, you will first produce your proteoses, and so you produce a proteoses and then a release of amino acids. And so your proteins are hydrolyzed to your proteoses, and then of course some amino acids are liberated. Okay, and then you have your proteoses to your peptones. And so when you hydrolyze your proteoses to your peptones, and so you will also liberate some amino acids. And so your peptones is further hydrolyzed to produce your peptides, and then still you have here the release of your amino acids and your peptides. And so your peptides here is finally hydrolyzed to produce your individual amino acids. Okay, and so that is your amino acids. That is your end. That is the end product of your protein hydrolysis. Okay, and so you have here in your hydrolysis, of course, you have there the addition of your water across the peptide bond, and so with the formation of your free amino acid. Okay, and your carboxyl group for each peptide bond will split accordingly. And so with that, hindi na po amino acid residues ang napuproduce mo, but then you already have your amino acids because and so your you already there is an addition of water. Okay, and so giving you already an amino acid. And so not anymore amino acid residues. And so amino acid residues, these are, and so when you join your individual amino acids to form a peptide bond, and so that is actually formed your amino acid residues. But then in hydrolysis, and so you already have your individual amino acids. Okay, and so this is the flow of the hydrolysis of your uh, proteins, and so with the use of your dilute acids and alkalis or your enzymes, with the use of your enzymes. Okay. Okay, and so your proteins, and so they give color reactions, and so they give color reactions with reagents, no? And so they give color reactions with your reagents, and so and so these color reactions are not specific to the protein, and so they are not specific to the protein, but and so that is because, and so they give color reactions because of certain groups so, that are present in your amino acids. Okay, and so again, and so your proteins, they give color reactions with your reagents, and so like your Sakaguchi reagent, your Hopkins color reagent, and the rest of your color reagents, no, for your color test for your amino acids, proteins, okay? And so these color reactions are not specific for the protein. But then, and so that is because to the certain groups of the component amino acids present in the protein, okay? And so other than that, and so your proteins will unite with some dyes also, like your picric acid, methylene blue, and others. And so also with that, okay? And so you have there. Okay, and so let's take a look at your individual test. And so you have here your first on the list, you have your Milon's test. And so your Milon's test, and so it uses your nitric acid and your mercurous nitrate, no? And so it uses your mercurous nitrate and nitric acid, okay? And so accordingly, and so when you add this one, and so when you add this reagent into a protein solution, and so the protein is precipitated as a mercury salt, no? And so the protein is precipitated as mercury salt. And then when you heat the solution already with a nitric acid and mercurous nitrate, and so it will, your salts, your mercury salt, and so turns from flesh to red color. Okay, and so if the protein contains tyrosine, and so it will give a positive test for Milon's test. Ayan, so if it does not contain tyrosine accordingly, and so it will give a negative one. And so what is present in your tyrosine that will give a positive result for this? And so the presence of your phenol group. Okay, and so the presence of your phenol group. Okay, and so your phenol group that is present in your tyrosine is responsible for the color reaction. And so in your Milon's test. And so with this, and so any substance that contain your phenol group will give a positive result to Milon's test. And so any substance that contains a phenol group will give a positive result to Milon's test. Also, any substance, any protein that contains tyrosine also will give a positive result to your Milon's test. And so, of course, the positive result here is a red color. Again, and so in your Milon's test, and so your nitrated tyrosine combines with mercuric ions to form a red color. Okay, and so the positive result is a red color for Milon's test. Okay, moving on. Your proteic test. And so it utilizes your concentrated nitric acid and concentrated sodium hydroxide. Okay, and so first, and so when you treat your protein solution containing when you treat a solution containing protein with concentrated nitric acid, it will turn into yellow. And then when you add your concentrated sodium hydroxide to neutralize the solution, it will change into orange. Okay, and so from yellow to orange with the addition of your sodium hydroxide. And so accordingly, and so your nitration of benzene, the benzene ring present in the structure of your amino acid is uh, the is responsible for this one. Okay, and so and then of course nitration of your proteins with the nitric acid will cause the solution to turn yellow. And then of course again, and so neutralization of the solution with sodium hydroxide will cause the solution to turn orange. Okay, and so with that, and so that is this color change here is due to the nitration of benzene ring present in the structure of your amino acids. Okay, and so what are your amino acids that contain your benzene ring? And so you have your tyrosine, your tryptophan, and phenylalanine. Okay, and so when you have Okay, and so when you have a protein that contains tyrosine, tryptophan, and phenylalanine, and so they will give a positive result for this test, for your santoproteic test, of course. And so a test, and so a positive result of yellow to orange. And so that is for your santoproteic test. And so it detects your benzene ring present in the solution. Your Hopkins Cole test. And so your Hopkins Cole test, and so this one, and so it utilizes your glyoxylic acid and concentrated sulfuric acid. Okay, and so glyoxylic acid and concentrated sulfuric acid. Okay, and so accordingly, ayan, so your glyoxylic acid here combines with your indole ring that is present in the tryptophan. Okay, and so it combines with your indole ring present in the tryptophan, and then this tryptophan, and so will condense with the aldehyde from your glyoxylic acid to form a colored compound. Okay, and so a positive test for this is a violet ring at the contact of two liquids. Okay, and so a positive test for your Hopkins coal is a violet ring at the contact of two liquids. Okay, and so you have here your glyoxylic acid and your concentrated sulfuric acid, and the positive result is your violet ring. Okay, and so your tryptophan, which contain your indole ring, and so will give a positive result for your Hopkins coal test. Okay, and so of course with your the proteins that contain tryptophan will give a positive test for this, and so uh your other substances that contain an indole ring accordingly will give a positive test for this lead acetate test. And so it utilizes your 10% sodium hydroxide and your lead acetate solution. Okay, and so you have here, and so this one, it tests your sulfur group, no? And so your sulfhydryl group or your SH group here. Okay, and so you have here, and so for your lead acetate, and so your treatment with your sodium hydroxide will split the sulfur to form your sodium sulfide. Okay, and so there is with this, and so there is a degradation of your SH, your sulfhydryl group, or your SS group in your amino acids. Okay, and so that is under strong alkaline condition, and so that is why you use your 10% sodium hydroxide here. And so again, in your lead acetate, there is a degradation of your sulfhydryl group or your SH, or you have the S group in amino acids, and so that is of course under a alkaline condition. Okay, and so with your sodium hydroxide, and so it will split your sulfur, and so your it will split your sulfur to form your sodium sulfide. Okay, and so you have there. And so, and then when you add the lead acetate, and so when you add lead acetate to the solution, and so it will give you a black precipitate. Okay, again, and so after the split of your split of your sulfur to form your sodium sulfide, and then you add a lead acetate solution, and so it will form a black precipitate, and so that is known as lead sulfide. And so there is a black precipitate due to the formation of lead sulfide. Okay, and so that is what are your amino acids that will test positive for your lead acetate test. You have your cysteine, and your methionine. Okay, and so these ones, of course, accordingly, they contain your sulfhydryl group, and so you have your sulfhydryl group present in them. Your Sakaguchi. Your Sakaguchi test, and so it utilizes your sodium hypochlorite, your alpha naphthol, and 10% sodium hydroxide. Okay, and so again, and so it utilizes your sodium hypochlorite, your alpha naphthol, and 10% sodium hydroxide. And so in an alkaline solution, and so a protein in an alkaline solution, a protein that contains an arginine, and so arginine, or yes, an arginine will give a positive test for your Sakaguchi. Okay, and so what is the positive test for your Sakaguchi is indicated by a red color. Okay, and so your sodium hypochlorite here, and so a component of your Sakaguchi test, and so it is the oxidizing agent. Okay, and so it is the oxidizing agent, while your alpha naphthol here, and so this one is the condensing agent. Okay, and so the reaction between the guanidonium group present in your arginine with alpha naphthol in alkaline solution will give a red color. Okay, and so with this, and so proteins that contain arginine will give accordingly a positive test for your Sakaguchi. Okay, and so and others, and so your guanidine, and then those that contain your guanidonium group will give a positive test for Sakaguchi. Okay, and so in the positive test here is your red color. Okay. And then you have your sodium nitroprusside test. And so you have your sodium nitroprusside. It utilizes your sodium 1% sodium nitroprusside and 10% sodium hydroxide. Okay, and so with this, and so your proteins that contain your cysteine will give a red color, no? It will give a red color for this one. And so in a dilute ammoniacal solution, no? And so your alkali, and so your alkali here, of course, that is your sodium hydroxide, will liberate your sulfhydryl group. Okay, and so your sulfur reacts with your sodium nitroprusside to produce a red colored complex. Okay, and so this one is a test for your cysteine. Okay, and so with this, and so we test for your cysteine. And so the thiol group that is present in your cysteine. Okay, and so why that, bakit cysteine lang ang nilagay ko dito? Because accordingly, and so your methionine, and so your methionine, it also contain a sulfhydryl group, no? And so, but then in your methionine, and so the sulfhydryl group present in your methionine, that is relatively difficult to break. Okay, and so the sulfhydryl group present in your methionine and so is difficult to break. Okay, and so note that in your cysteine, and so you have SS only, and so you need your thiol group that is present in your cysteine, and so that will give a positive test for your sodium nitroprusside. Okay, and so you have here your SH dapat. And so you also have an SH, sabi ko nga in your methionine, but then, and so it is uh, relatively difficult to break. Again, and so it's difficult to break. Okay, and so that is for your sodium nitroprusside test. Okay, and so moving on, and so this one is your structure of your glycine. And so this is often considered as a non-essential amino acid, no? Ayan, so because of course the body can synthesize it, and so it is involved in the synthesis of important biomolecules like your protein, such as your proteins, of course, and so uh, deoxy deoxyribonucleic acid and your bile salts accordingly. Okay, and so you have here the structure of your glycine. Moving on, you have here your cysteine. And so this one is a sulfur-containing uh, amino acid, no? And so it is accordingly unique among your amino acid because it contains a thiol group. Okay, and so it contains a thiol group. And so this one, your thiol group present in your cysteine, and so is important for the formation of disulfide bonds. And so important for the formation of disulfide bonds, and so which contributes to the stabilization of your protein structures. And so as what we talked last time in the other video, no? And so this one is your cysteine is a precursor of glutathione, and this one is a powerful antioxidant in the body, no? And so that is for your cysteine, and so it contains a thiol group. Then you have your tyrosine, and so is this one is classified as an aromatic amino acid, and so note that it contains a benzene ring, and so it contains a benzene ring, no? And so this this one makes it this benzene ring accordingly, and so make it structurally different from non-aromatic amino acids. Okay, and so it has a benzene ring, no? And so this one, your tyrosine is a precursor to neurotransmitters like your dopamine, norepinephrine, and your epinephrine, no? And so that is for your tyrosine. And this one is a precursor for your hormone thyroxine also. Okay, moving on, you have your proline. And so your proline is unique accordingly, and so its side chain forms a cyclic structure, no? And so it has a cyclic structure, and so you have here, and so it creates an imino group instead of amino group accordingly, and so it creates an imino instead of amino group. And so this the cyclic structure, and so accordingly the cyclic structure of your proline, and so restricts the flexibility of the polypeptide chain in which it is present. And so the cyclic structure again, and so present in your polypeptide chain, sorry, the cyclic structure, and so of your proline, and so it restricts the flexibility of the polypeptide chain, and so in which this proline is present. Okay, and so this one, and so of course giving you a rigid structure, hindi na siya flexible, no? And so this rigid structure of your proline is important accordingly, and the stabilization of protein structures, and so that particularly in the turns and loops of your proteins, no? And so that is important in your beta and your beta pleated sheath, and so those items in your alpha helix, no? And so that is for your proline. Okay, moving on, and so you have here your what do you call this, you have here your summary of the test that you did, no? And so you have here for your sodium nitroprusside, and so your sodium nitroprusside, your lead acetate, sodium nitroprusside test, your lead acetate test, and so your Hopkins Cole test, your Santoproteic test, and so your Sakaguchi test, and your Milone's test, and so all of which will give a positive test for your albumin, I mean a positive result. And so when you test your albumin, egg albumin, and your milk with the following, and so it will give a positive result for this. And so what is the important thing here is, of course, to note what are the components of your milk and your egg albumin. Okay, and so what are the components of your milk and your egg albumin? And so with that, and so they contain the amino acids that will test positive for this test, and so thereby giving you a positive test for each of the tests here. Okay, and so moving on, and so your nitric acid stains are challenging to remove, no? And so by simply washing, because your nitric acid is, this one is a strong oxidizing agent. And so this one, your nitric acid is a strong oxidizing agent, and it can cause chemical changes in the substances that it comes into contact with, no? And so your nitric acid is a corrosive and reactive chemical that can oxidize, degrade, or even modify the structure of materials, including your fabrics and your surfaces, even your skin. And so it can burn your skin. Okay, and so you have here with this, and so it is difficult accordingly to remove the stain, and so because your nitric acid, and so it can accordingly penetrate, and so you're deeply, and so it can penetrate deeply into your what do you call this, stain, no? And so you have there, and so your nitric acid can penetrate into your pores, and so nitric acid, being a corrosive substance, can penetrate into the pores or interstices of materials, and once it has already penetrated deeply, and so a simple wash, and so it's not efficient, it's not enough to remove the stain of your nitric acid. Okay, and then it forms a also that contributes to the difficulty of the removal of the stain, and so it reacts with certain substances to form insoluble compounds, and so that are difficult to remove in normal washing. Okay, and so and these compounds adhere strongly to the material, making it challenging to remove. Okay, and then of course, and so your nitric acid, and so nitric acid, and so may what you call this, oxidize again or break molecular structure of a material, and so leading to a discoloration again, and so that is for the staining of your nitric acid. Okay. And then you have here a summary of your what you call this, of your a summary of the different tests that you did actually here. And so you have here your sodium nitroprusside, and so I added here your Sullivan's test, and so you have actually a lot of tests for your amino acids. And so other than that, you have your Fullin's reaction, and so you have your Lieberman's test. Also, you have your poly reaction, and so but then for this, and so we only have this. I only added your Sullivan's test here, and so your sodium nitroprusside, and so the positive result is cysteine, and the amino acid is cysteine, no, and so that is the component that is being detected here is your sulfhydryl group or your thiol group or your thiols, no, your lead acetate, and so a black precipitate. And so you have here your cysteine and methionine, and of course you have your sulfhydryl group. Your Hopkins coal, and so violet ring at the contact of two liquids. And so you have your tryptophan as the positive, I mean as it will give a positive test for your Hopkins coal, no? Your xanthoproteic, and so yellow to orange, and so you have your tyrosine, tryptophan, and phenylalanine are the representative amino acids for this test, no? And so, and of course, that is due to the presence of benzene ring. Your Sakaguchi, you have your red color, okay, and so your arginine and your guanidonium compounds. Okay, and so your arginine and your guanidonium compounds, no? Your guanidonium group. Okay, arginine and guanidine, no? And then you have your Milan's test. And so your Milan's test, and so this one is what they call this, a test for your, actually for a glycoprotein, no? And so if a protein solution is accordingly treated with a Molish reagent and layered with a, what they call this, and you added, sorry, and you added a, uh, sulfuric acid, concentrated sulfuric acid, and so a violet ring will develop, no? And so if again a glycoprotein is present, no? And so glycoprotein, and so this one is for your the carbohydrate present in your protein, okay? And so your sorry, and so it's Milan's, no? And so it's Milan's test. Okay, and so your Milan's test, and so will your Milan's test, and so it will give a red color. Okay, and so I was talking of Molish test kanina. Okay, and so your Milan's test, and so you have your tyrosine, and so as the representative amino acid, that will give a positive result in Milan's test, no? And you have here your phenol group. Okay, and so you have the phenol group, and then you have your Sullivan's test, and so your Sullivan's test, and so you have your red color. And so in your Sullivan's test, and so in the presence of a strong reducing agent like your sodium hydrosulfide, and so your cysteine in alkaline solution, and so still, and so gives a red color, no? And so with the sodium 1,2-naphthol for sulfonate. And so this one can be used, this Sullivan's test here can be used for the estimation, quantitative, no? Quantitative estimation of cysteine and cysteine after they are reduced. Okay, and so that is for your the test that you did in your activity 19. Okay, and so sorry for this. And so it should be Milons. I was talking of Molish kanina.